Basically, the study describes how we used mass spectrometry as a way to find monoclonal proteins in patients with various plasma cell disorders. So my name is Surendra Dasari. I am a PhD scientist at the Mayo Clinic. We're here today to talk about an article that will be appearing in Mayo Clinic Proceedings, and the title of the article is Detection of Plasma Cell Disorders by Mass Spectrometry, a comprehensive review of 19,523 cases. So most notable plasma cell disorders are like multiple myeloma and light chain amyloidosis. And for nearly 50 years, gel-based electrophoresis is the main method that is used by the clinical labs to screen for M proteins or monoclonal proteins. And while they are really good methods, they do suffer from lack of sensitivity and specificity. And what I mean by that is, for diseases like light chain amyloidosis, when the M protein is really small, sometimes it's really hard to see them on gels. And specificity, lack of specificity, what I mean by that is, like if there are patients who are treated with these modern monoclonal therapeutic antibodies, and you look at their serum sample, sometimes they would have both the pathogenic M protein and the monoclonal M protein present. And it's really hard to distinguish on a gel what is a therapeutic antibody the patient was given versus what is actually the original pathogenic M protein that is in the patient. So what we've done is we developed this new method that uses mass spectrometry, which is a very high resolution method to get around this sensitivity and specificity issues. And a great example is that, when we look at patients who've been treated with therapeutic monoclonal antibodies, we can clearly see, just based on how the mass spectrum looks, we can clearly distinguish between the M protein that the patient has produced versus the therapeutic monoclonal antibody. And the other advantage is there are actually other more advantages to the method. Another advantage is that the new method can reveal features of M proteins like light chain glycosylation, which are actually tied to worse prognosis. And actually, the light chain glycosylation is also a marker for AL amyloidosis, which there is really no good marker for AL amyloidosis up until now. So apart from the clinical advantages of being more sensitive, more specific, and detecting these pathogenic features of these M proteins, the new method is also highly scalable in a sense that it is actually 30% more efficient than the current gel-based electrophoretic methods. So what makes the study so unique is that we actually implemented this method at the Mayo Clinic, and we ran about 19,000 samples. And then we actually looked at the clinical characteristics of the monoclonal proteins and the patients that turned out to be positive. We never actually missed a M protein. Plus, we found all of these patients with therapeutic monoclonal antibodies in them. We found patients with light chain glycosylation. And overall, the process was also more efficient, meaning we were able to turn over more results per lab tech. So overall, the process was very, very rewarding. And not only for a lab, but also from a patient perspective, it was actually a very great method. So most of you may know that the Mayo Clinic has a rich history of innovation in plasma cell disorders, with starting with Dr. Bob Kyle. So we were basically following his footsteps. So it makes it a little bit better and easier for us because of the biobanks that are accumulated and the clinical knowledge that's been accumulated, and just the history of innovation around the plasma cell disorder space makes it easier for us to take a new unknown test and put it in the clinic and give the result to a hematologist and actually use it for patient care. As of today at the Mayo Clinic, we are not running gels for routine screening of monoclonal proteins. We now actually use the mass spec method for doing the screening. Whenever we find these pathogenic features like light chain glycosylation, we start thinking about how do we treat the patient differently? How do we follow them up differently? So in the clinical area, the having more sensitivity, having more specificity is definitely more advantageous. What does it mean for patients? If we can find a clone, a pathogenic clone that, like in amyloidosis, if you cannot see it on a gel, but you can clearly see it on the mass spec, and it persists, and the patient has elevated light chains, free light chains, then you know that you have to act on the patient. Again, you find a patient with a glycosylated light chain, then you know that you need to look at the patient a little bit more carefully. When you see a treated patient with a monoclonal therapeutic monoclonal antibody, but the original clone is also there, then you know that you may have to track the patient a bit more closely because the original pathogenic clone is still present. So all of these will add up to benefits to the patient because the clinical care is more personalized, it's more tailored. Obviously, we have just started implementing this clinically, and there are many more steps to make it widely available to the people outside of the Mayo Clinic, which is happening as I speak. A lot of work is being done, and we also have wanted to go into the areas of minimal residual disease because, like I said, mass spec is more sensitive, so we can see clones, lot smaller clones than you would see it on a gel. So we are also interested in going into this minimal residual disease and define that a little bit better so that we can increase the response rates for these patients. And finally, I would like to invite you all to read the article for more details. And if you have any questions or comments, please feel to reach out to us. Thank you very much.
We hope you found this presentation from the content of our website valuable. Our journal's mission is to promote the best interests of patients by advancing the knowledge and professionalism of the physician community. If you are interested in more information about us, our homepage is www.mayoclinicproceedings.org. There you'll find access to information for our social media content, such as additional videos on our YouTube channel or journal updates on Facebook. You can also follow us on Twitter. More information about healthcare at Mayo Clinic is available at www.mayoclinic.org. This video content is copyrighted by Mayo Foundation for Medical Education and Research.